morphology in a poem?
Woo! Hi everyone, I'm Claire, and like Naomi said, I'm going to be talking about the factors that influence leaf morphology for a cloud forest palm. This one is Geonoma brenesii. So, before we start, I want everyone to think about a leaf. Any leaf, just think about a leaf in your mind. What shape is it? What size is it? What color is it? Visualize this leaf. Does it look anything like any of these? These are some of the thousands of different morphologies that leaves can have. So leaf morphology is the shape of a leaf. So morphology can be long and slender, it can be compound, it can be many different things. And these shapes are not stagnant over time. They actually can change in response to factors. So some factors that they change in response to are light exposure, humidity, elevation, different things, rainfall. And the fact that they change, that is termed leaf plasticity. So really what I'm talking about today is the leaf plasticity of Geonoma brenesii. So Geonoma brenesii is a palm located in the cloud forest nearby. So they're found between 1,000 and 1,600 meters. This is taken right next to the symposium biologico, right near where they're found. They're endemic to Costa Rica, and specifically in the Tiburon and Central Mountain regions, which means you will not find them anywhere else in the world, only here. Why I'm interested in them is because their leaf morphology is very unique based on all my observations. So, I collected leaves. First I was hiking, I saw that they vary so much, so I collected leaves and I took photos, and I found that there are symmetrical and asymmetrical leaves. The symmetrical are, starting here, we have 2 leaflets, 4 leaflets, and 6 leaflets. So this right here is the center vein, this is the rachis, and you have 1 leaflet on either side. This one you have 2 on either side and 3 on either side. As you can see, they're symmetrical, they're the same on both sides of the rachis. Then I found some that were asymmetrical. So we have 5 leaflets, 5 and 7. They can get up to 15 leaflets per leaf, which is quite a lot. But even among the ones that are both 5, you can see that the orientation is very different of the leaflets. So this whole thing together is 1 leaf, and each one of these is a leaflet. So that led me to my questions, which are, why is this happening? Why is there so much variation in the leaves I was finding? So I have number of leaflets, that's what I want to know what was being affected, and these are all the variables that I was considering. So first, I was considering crown width, and then crown height. So crown width is the actual width of the whole plant from tip of leaf to tip of leaf. Crown height is the same, it's from the bottom of the stem as it meets the soil up to the top of the highest leaf. Stem circumference, that's below the lowest node. Number of leaves of the whole plant. Percent canopy cover, so that's how much sunlight is actually reaching the plant. And the elevation. I also decided to use very similar variables to determine if it had any effect on bilateral leaf asymmetry. So again, this is the rachis. Bilateral leaf asymmetry will mean left and right side asymmetry. So these ones are symmetrical bilaterally, these ones are asymmetrical. And I did the same variables, except I added one. I added leaflet asymmetry. So leaflet asymmetry is when the number of leaflets on either side is not symmetrical. So this would be a leaflet asymmetry of 1 because it's 3 minus 2. Same with this, 1 and 1. So my methods were, first I had to locate the species. I used Eladio's help for this. He is the expert, and we were able to identify and locate the number of these Geonoma brenesii. And then for each plant we measured the elevation, the percent canopy cover, so again the sunlight, number of leaves, stem diameter, canopy width, and canopy height. And then for each leaf, for each plant, we picked out 6 leaves to measure. 2 from the top, 2 from around the middle, and 2 from around the bottom, just to get some variation in our measurements. And we measured right here what you see. So this line right here is the rachis, we measured that. We measured this, which is the primary leaflet, so the size of the primary leaflet, the length. We measured the number of leaflets, so here we have 2. And then we measured the space in between, which is a little space right here. If they were symmetrical, we only measured for the left side. If they were asymmetrical, we measured both sides so we could calculate asymmetry. Results, so first we used RStudio, or I used RStudio to calculate the multiple linear regressions to figure out if there's a correlation. And I also calculated leaf asymmetry using the area here. So basically I would do radius minus the length of the space in between to get the length of the total leaf area for one side, multiply it by this length, and that would give a rough area of the left side. And then I would do the same for the right. If it's symmetrical, the difference is 0, but if asymmetrical, then we got a value. So total, I collected 3,380 measurements, which took quite a long time. Eladio helped with all of that. 48 plants in total, and we measured 282 leaves. Of all of the leaves we measured, 56.25% were asymmetrical. So of the 282, 153 were asymmetrical, so more than half. For all the plants we measured, almost 90% had one or more asymmetrical leaves. So that means only around 10% were fully symmetrical on all the leaves. The max number of leaflets we found was 8 for one side, so 15 total for the whole plant, or for a whole leaf. And the maximum elevation was 1,750 meters. So, do they correlate? This was the question, and we got an answer. After doing the regression, we found that number of leaflets, stem circumference determines or correlates with number of leaflets. So this positively affects number of leaflets, which means as stem circumference increases, number of leaflets also increased. We found for bilateral leaf asymmetry that as crown height increased, so did bilateral leaf asymmetry, same for leaflet asymmetry and elevation. So why might this be? Question 1 was if number of leaflets is affected by anything. We found stem circumference affects number of leaflets. This could be due to, okay, this is a German word, called, or it's pronounced Wachstum, or something, strengthening growth. It basically means that as stem diameter increases for a plant, specifically monocots, more so monocots than other plants, that leaf size and complexity increases. So if leaf size and complexity increases, it makes sense that the number of leaflets also increases. For the other question, what affects bilateral leaf asymmetry, we found crown height, leaflet asymmetry, and elevation affects bilateral leaf asymmetry. So again, crown height, just like stem circumference, crown height also is a part of Wachstum, and so again, leaf complexity and size corresponds to higher crown height. So the taller the crown, the more variation there is in the leaf, and so then this could be deduced that it's also asymmetrical. Leaflet asymmetry affecting bilateral leaf asymmetry can be explained by the total area of both sides. And then I was not able to find any reason why elevation could affect bilateral leaf asymmetry, so this is unclear and could be studied in the future. My conclusions, stem circumference affects number of leaflets, so we found that. We found that bilateral leaf asymmetry is affected by crown height, elevation, and leaflet asymmetry. So these are all correlations that are statistically significant. For further study, when we were examining these plants, there was a lot of very slight crossover or very similar morphology of the plants, and so it's a little bit unclear if we were studying more than one species perhaps, or one species that was sort of going into two species. There's I think 3 studies total on this species overall, so a lot of information is lacking. So if a taxonomist came and examined these, that would be great. Also, elevation and leaf asymmetry correlations, still no idea why this happened. That would be another great area to study. And another thing that could have been done in this study or for a future study that we weren't able to do is there's an actual asymmetry index that can be used to measure asymmetry. That gives a very precise number, more precise than what we found, so that could have helped with our calculations. Okay, I'd like to thank Eladio Cruz Leton for all of his help. Hay una cosa que me asusta, que me asusta y es son insectos. Y Eladio me ayudaba mucho porque cuando medíamos las plantas y había insectos, él hacía todo y no podía hacer sin usted, Eladio. Muchas gracias. Y también thank you to Federico for being so kind and knowledgeable and helpful. Wherever you are, there you are. And thank you for Naomi for everything, for all the answering all the questions all the time. Okay, that's all. Any questions? Or what are your questions? Yeah.
So, for the area measurements, for the asymmetrical leaves, which side normally has more area, the one with more leaflets or the one with fewer leaflets?
The one with fewer leaflets has more area. Yeah, that's what we found, so, or what I found. So this side, the left side here, would have a greater area than the right side. But, yeah, yes. We took it, basically with the absolute value, so in the end it didn't matter which side, like left or right was greater, we just found the difference between them. Yes?
Do you think there's a purpose for the asymmetry?
It was a little bit unclear. One thing I found in my research is that a lot of plant, like leaf plasticity, has to do with, like I mentioned, like a bunch of factors, external factors, like light and other things. So it's possible that they're adjusting to micro abundances, like micro differences in light, versus like one whole plant could be different from a whole other plant, but that's like expected because they're in different spots. But even if one little spot in the canopy is exposed to one side of one leaf, then it could cause that, although it's a little unclear. Yes?
How consistent do you think their findings are when it comes to leaves in other biomes?
In other what?
Biomes, like not necessarily rainforest, but like in maybe tropical climates or other tropical climates or even seasonal climates.
Yeah, I'm not sure. I know that the concept of the German long word, that applies to more than just in cloud forests or tropical rainforests. So that has to do with leaflet complexity or leaf complexity. So asymmetry could also be explained by that in other like life zones. Although this plant is only here in these cloud forests, so in terms of this plant, I don't know, because it wouldn't be located elsewhere. Any other questions? Thank you, Claire.